Hi, everybody. Welcome to Tuesday Talks at Cure. I'm Seema Kumar, and today we're going to chat with a Cure resident company that is developing what hopes to be a groundbreaking regenerative treatment for people diagnosed with Parkinson's disease. Nader Haleem, a biologist by training and the CEO of Innervace, is here to tell us how his company is developing implantable brain pathways that can reconnect areas of a brain affected by Parkinson's, which, as we know, affects millions of people worldwide. I'll also discuss with Nader, who is a first-time CEO, what it takes to launch a new biotech company and how he splits his time between the lab and running his business. Welcome, Nader. How are you?
I'm great. Thank you for having me, Seema.
Yeah, so much to discuss. So let's dive right in.
Okay.
And before we get to you and Innervace and forming of the company, let's talk about your background. So you were in academia, and you also have worked in pharmaceuticals and biotech companies. So tell us a little bit about your background.
Yeah, I started out, as a lot of folks do, in academia. I did my PhD, and as I was approaching the end of my PhD, I knew that being a bench researcher, bench scientist and academic wasn't really necessarily my career path. And I think that's really a common trend that we see with folks now in graduate school. Very early on, everyone went into academia. But when I was finishing up, and really much more common now, folks have other opportunities to move into industry. So after finishing my PhD, I went into consulting for a couple of years, really focused in on the life sciences space, managed to get a great depth and breadth of functional areas within the pharmaceutical industry. So I've worked in commercial space, I've worked in policy, pricing, research and development. Did that again as a consultant for about 6 years, a couple different firms, and then transitioned to Genentech, Pfizer, a few other small biotechs, and then in 2020 I started at Innervace. In 2022 actually came on full-time as CEO.
That's great. So, I mean, how did all of that experience, right, working at NIH and Pfizer and Genentech, prepare you for launching Innervace? What was it about those experiences that prepared you well?
Yeah, I think it was really the diversity of experience that really helped me, working on the bench. I am now at the bench again. I had over a 10-year hiatus from the bench, so it's nice to be back in the lab, working with my hands, but really meeting different people, understanding how a pharmaceutical organization, generally mid or large pharmaceutical organizations operate, what pharmaceutical industry is looking for. So, most biotechs are ultimately acquired by a larger pharmaceutical company. So really having that eye on what is the path for a therapeutic to be developed, and what is industry looking for? What are the regulatory hurdles? What are the overall policy trends that we should keep in mind, whether that's pricing or government reimbursing when we're developing a therapeutic, and really integrating that information earlier and earlier into the R&D cycle. So, the product that we have, of course, meets a patient needs and is well prepared for commercial launch.
Speaking of products, and talk about Innervace, you are actually developing a technology that originated in academia, right?
Yeah.
One that uses what you call a preformed, anatomically inspired implantable tissue pathways. Even as I'm saying it, seems to be like science fiction, right?
Yeah.
This is what it does, is it replaces the connections that are lost in the brain in degeneration in patients with Parkinson's. I mean, that's
right.
like awesome. How does it work?
Right, so the idea is in Parkinson's disease, there's a group of cells, they produce a neurotransmitter called dopamine, which most people have heard of. It sits within the deeper structures of the brain and it projects to a more forebrain structure. Those cells die in patients with Parkinson's disease. That decreases the amount of dopamine in this forebrain structure called the striatum. And we've really known the biology, the underlying cell biology of Parkinson's disease for almost a century now, when it was described by Dr. Parkinson. So we understand the biology. And because there is just a group of cells that have degenerated, a very defined pathology, unlike Alzheimer's. So, Alzheimer's is the most prevalent neurodegenerative disorder. There are changes throughout the brain. In Parkinson's disease, it really originates again with one group of cells dying. So because we understood that population of cells, we understand the pathology quite well, the idea of a regenerative medicine approach of replacing those neurons or the dopamine that is lost by those neurons being degenerated has been an idea that's been in the field for about 40 years. So some of the very first trials started about 40 years ago. There were some interesting results, some interesting signs of efficacy, generally quite safe. So the concept or the idea of regenerative medicine for Parkinson's disease is really a decades-old concept. What we plan to do, and there really is a resurgence in regenerative medicine because of developments in stem cell biology, which has allowed us to generate pure populations of dopamine neurons. Our approach is a little bit different, where we're really trying to recapitulate that circuit. So the brain really is about circuit. Neurotransmitter is produced in one area, released in another area. The idea we have is, can we reconnect these two different regions of the brain? Because there are other pathological changes that happen later in Parkinson's disease. So we really want to take these two, distal brain regions, reconnect them. Essentially like a tissue transplant is one way this could be described, and hopefully be a disease-modifying therapeutic, the opportunity maybe even to reverse some of the symptoms of Parkinson's disease.
Wow, that's just amazing. I mean, because current standard of therapy, what is it now? Just a medication, right?
That's
right.
Is it
just plain old dopamine, and
L-DOPA.
it's just
yes, a
L-DOPA,
precursor
right?
of dopamine. Yep.
Yeah, a precursor of dopamine, which is L-DOPA,
Yep.
that's given to patients. So
Yeah.
you can continue to replenish the dopamine, but ultimately the real solution is to have neurons that can actually generate dopamine and send signals to the front of the brain. Right?
That's right. So, current treatment is really around symptom management. It does really well for patients, particularly early on in the course of disease. As the disease progresses, unfortunately, patients may lose responsiveness to L-DOPA. L-DOPA in and of itself has side effects, and it doesn't change the total end course of the disease. So it is really symptom management. There's also deep brain stimulation, which can help with symptom management. But really what we want to do is disease modifying, change the trajectory of the disease. Really, that is our hope. And even another hope would be to stop or reverse disease. So, we're focused in, and there are a number of other companies on the regenerative medicine approach. There's another group, and I've worked on some of these projects in Big Pharma, which is also trying to delay progression of the disease. Unfortunately, when a patient is diagnosed with Parkinson's disease, they've lost the majority of the neurons that produce dopamine. So without better screening tools to identify patients earlier, prevention and halting the course of the disease is a little bit of a challenge, but we're making some good headway.
Sometimes it may be a little too late and too, short. I mean, yes, we will take whatever we can.
That's right.
But, it's probably by the time you notice the symptoms, it's already a little bit far gone.
That's right. You've lost a lot of those neurons. There is there's some great biomarker work that was funded by the Michael J. Fox Foundation, and as that biomarker kind of grows, our understanding grows, and we can get that biomarker out to patients, I think there's an opportunity to work on more preventative trials. Those are generally larger, more costly, but really any angle that we as a scientific community and industry can work on this disease to help patients with this disease, I think all approaches are needed to make an impact with patients with Parkinson's.
So let's make sure we understand this correctly, especially our audience and me included. That is, so this regeneration of these neurons, right? It's not that you're actually transplanting neurons into the brain, but you're also rewiring it so that the circuitry also works. So how does that happen?
Right, so we actually are implanting neurons in the brain. So we are using stem cells. We are taking those stem cells, so it's kind of core biology, differentiating those into dopamine-producing neurons. We're then putting those in really what's the end of a tube. And then we're growing those neurons out really kind of towards the length of the tube. And that would be done in the laboratory setting, in a manufacturing setting, in a controlled setting. That implant is then taken and would be neurosurgically delivered so that the neurons sit in the region of the brain where they would normally reside, and one end of the tube, and the other end of the tube terminates in that region where the dopamine is released. And then we really let the idea
it's almost like a stent.
It's very similar.
Yeah.
So it's most likely it has elements of a device, but the main kind of active ingredient is the neurons, but we have to have them really encased in what we call a scaffold. And when the scaffold is implanted, then we really let the neurons do what they do. So they form connections on either end of the tube, and we hope that that would recapitulate the circuit that's lost within patients with Parkinson's disease. So we basically have the setup, the implantation, and then let biology go.
Yeah, this is, this sounds really wonderful, especially, I mean, given how frustrated families and patients with Parkinson's are. It's been a long time, right, that we've been kind of dealing with this disease, and it's phenomenal. So many people suffer from this, and technologies like yours could potentially offer not just a delay in progression of the disease, but ultimately actually resolve the disease, or even, dare we say the word cure?
That would be our hope, absolutely.
Yes. That's great. That's wonderful. Yeah. So, switching gears a little bit, right? I mean, this is just incredible science, and you've been working on this, and it's almost seems like a drug-device combination, right? And that's when I'm sure you're thinking about, the path forward. But in the beginning, as what made you actually start a company with this? And you were a scientist, I mean, and what advice would you give for somebody else who's sitting on incredible science like this and who's looking to start a new biotech? What are a few things you learned, since Innervace was founded, especially on managing science and business, and on making sure that the science that you have in the laboratory can be translated in the commercial setting? Absolutely.
So the concept, the idea was not my idea. The technology, the idea of reconnecting regions of the brain that have been disconnected, our scientific founder, Casey Cullen at the University of Pennsylvania, this was really his brainchild. There was a group that was looking to do a seed investment, and they had asked me to, take a look at the company, think, what did I think was their potential? And I saw really what was a very transformative opportunity. And what you mentioned sounds like science fiction, but the science has really caught up. So hopefully that science fiction can become science fact. There's always that interface where we reach, and then we reach and we actually achieve something. And to me, again, the advances in stem cell biology, advances in complex manufacturing, cell therapy as a field has really exploded. So I thought this was an amazing opportunity. I worked with Casey for a number of years just out of his lab. We then went through, we were fundraising, raised a Series A, and that's when we took Innervace, again, really went off to the races, joined the Cure.
That's wonderful. That's really great. I think because many academics, like you said at the top, are actually looking for ways to, translate their science. Partly because when you translate the science and bring it to, to patients, there's an immense amount of satisfaction to have, made the world a better place and bringing forward cure. We, of course, are proud to have you here as a resident at Cure. And you are an important part of the ecosystem, and it's companies like these that we love, to be part of the ecosystem. So how is that working? How is it to be part of Cure, and what has it been like?
It's been fantastic. Have to be in a building dedicated to, the advancement of health is phenomenal. To be with folks who are like-minded. We have a partnership with a medical device incubator within the building. That has been our lab space. We receive engineering support. We are not developing a traditional therapeutic. It is a therapeutic plus. There are device elements to it. So the diversity of expertise that you need is really quite large. And having folks within the building, have those informal conversations, those formal collaborations and partnership has really been helpful. And also being in Manhattan and in New York City. I've lived in New York City for almost 15 years now, and it's great to have a center that is dedicated to the life sciences, because we sit in a very rich community with Columbia, NYU, Hospital for Special Surgeries. There's just an amazing, diverse population of patients. So it's really a great experience.
Wall Street and the investment
Absolutely,
community.
a lot of investment houses,
Yes.
backers. So it really, New York is just a great, obviously very eclectic area, and it's great to have a building dedicated to the mission of, companies looking to have an impact and make an impact in healthcare.
What's one piece of advice you would give to a first-time CEO or to somebody in academia who wants to take that leap?
Yeah. My first job, one of my jobs at Pfizer was to find academic technology and translate it into industry. The first thing is to be comfortable knowing what you don't know. We all don't know many things, and we're all developing things. So asking, looking around, not trying to be the subject matter expert. I think you need to pull on a diversity of folks. So, really being open-minded, to me being mission-driven. My uncle has Parkinson's disease, so I've seen the devastation of the disease. There are millions of people who could be helped and impacted. So, being drawn to the mission of the company, I think is really important. Probably the last thing is maintaining that scientific rigor. It's, while we have passion and we need to have faith in an idea because an idea is untested, really holding to the rigors of, true science. Most drug development efforts fail, and to be cognizant of that and to fail fast, but to do everything you can to push your concept, your platform, your therapeutic forward.
That's great advice. My father-in-law had Parkinson's, lived with Parkinson's for a long time before he passed away. And we know that this month is Parkinson's Disease Awareness Month.
Yeah.
What is your message to patients and researchers today?
Yeah, I think, the message to patients is, we are, as a community, working on the disease through many different approaches. I think, there's neuroscience to me, neurodegenerative disorders, as we all age and as we live longer, we all have a greater chance of developing neurodegenerative diseases. And these are some of the most devastating diseases, ALS, Parkinson's, Alzheimer's, where we really don't have a lot of treatment options. So that has not gone unnoticed. And a lot of us are really focused on trying to bring many different therapeutic interventions that are symptom management, that are disease modifying to patients. So really hope that within this decade, we'll see some transformative therapies that are approved for patients.
That's great. Well, thank you so much, Nader. Thank you for joining us today. This was an absolutely wonderful conversation.
Great.
And we wish you all the best. And, we're rooting for you because we need to find a cure for Parkinson's. So thank you for joining us. It was an absolute pleasure.
Thank you.
And thank you all for joining us, and see you all next week.